Okay, so today we're going to talk about DNA replication, or the most important copy machine in the world. So first, let's talk about what we're going to actually discuss today. First, we're going to talk about a little review of what we remember of the structure of DNA. And then we're going to move on to what exactly we're copying, when we're copying it, and why we even bother doing this. Why does this even happen? And then we'll talk about how it actually happens, and then a few other little details that go along with that. So first, we're going to do a quick matching game, a quick review game for the nitrogen bases that we discussed the other day. If you recall, you've got the four bases of DNA, and there's RNA has one different base in it that matches up to the DNA molecule. And what we're going to do is on Jamboard, we are going to you've got this square in the middle of bases, and you're going to match the corresponding bases, base sequences that I have here. And you may need to spin them to make them match, and just pay very close attention when you're doing it. There's a few tricks. I tried to trick you here, so go ahead and do that. All right, that was quite impressive. You guys nailed that really quickly. Now here's the answer. Now I know, like I said, I tried to trick you here in a couple ways. One is that on this sequence here, I threw in a few RNA bases, and then on these sequences, they're actually the same sequence with just the primes reversed. So you can see this one, in order for it to fit, needed to flip over the other way. So it looks like it doesn't fit, but it actually does. If you flip the 3 to this side and the 5 to this side, spin the whole thing around, you'll see that it does. You saw that it does match. Now I know that 5 prime to 3 prime direction is a tricky concept, and that's something that's really important for DNA replication. We're going to talk, so hold your questions for now, because I know you have questions about that. I certainly did, and we're going to talk about that in a few minutes. So let's review quickly. DNA, it's a nucleic acid molecule, and it's made up of 3 basic parts. You've got the sugar phosphate backbone. That's 2 of the parts. One is the deoxyribose sugar and the phosphate group, and that's what makes up this sugar phosphate backbone. And that's kind of the vertical parts of the ladder, if you will, that go up and down. And then the rungs across are those nitrogenous bases that we just matched up. So the adenine, thymine, cytosine, and guanine, those are the bases. So again, we need to remember that the 2 sides run in a anti-parallel direction. That's really important here. It runs in that anti-parallel direction. So you've got your 5 prime end to your 3 prime end on this side, but then on this side it's the 5 prime to 3 prime. And again, we're going to talk about that in a few minutes, a little bit more detail. So why are we bothering to do this? Why does DNA need to be replicated? Well, of course, it is the blueprint for all of our cells. It tells our cells and plant cells and bacteria cells and all the different cells that make up life what kind of cell they are, what their function is. So it's obviously important when that cell divides that the new cell has that information. But in addition to that, DNA replication we can use DNA replication for everyday purposes, for everyday real life applications outside of just within our cells. This idea of this technique called polymerase chain reaction, it's used when you have a little DNA and you need to make more of it to be able to test it. So they use it in forensics, in crime scene investigations, and we use it in laboratory testing, which is actually very topical because the most accurate, most used test for COVID is a PCR test in which they take the RNA. So I'm not going to go too in depth here because we're going to talk about this in a few days. PCR we'll talk about PCR more in depth. But COVID is an RNA virus, so it doesn't actually have DNA. It only has its genetic code is only RNA, which again we talked about the other day is a single strand as opposed to the double strand of DNA. So what happens is that we can take the RNA of the virus and we can purify that, and using an enzyme called reverse transcriptase can convert it from RNA to DNA, and then using a process, a heating and cooling process, can then split that DNA back in half and then replicate it millions of times. And with that, we can then test and see if a sample taken out of your nose when it feels like they're scraping your brain, we can see if that sample contains this DNA that we created from the original RNA. And if it does, then you know you have COVID. If you don't, if it's not there, then you don't. But we'll talk about that a little bit more in depth later. So when are we replicating? When is this whole process happening? Well, if we recall, we talked about the cell cycle, and the cell cycle involves interphase, and then the mitotic phase where the cell splits. So you can see most of the cell's life is spent in interphase, and there's the three parts to interphase. There's the G1, there's S, which is DNA synthesis, and then there's G2. So G1 and G2 are both cell growth. The S phase is DNA synthesis. So where do we think DNA is replicating? At what part of the cell cycle do you think it's replicating? Yeah, right. DNA synthesis. So that's where this actually occurs. So the cell grows, and then we copy the DNA over, the cell grows a little bit more, and then the cell starts to divide in mitosis, and then it actually splits in cytokinesis, and that's when the two daughter cells are formed. So now that we know when it's happening and why it's happening, we need to start talking about how it happens. This is where it gets a little complicated. So the first thing I'm going to mention, and then we're going to go into more detail on all of these, is the enzymes that are involved. And they're kind of the key players. They're the main players in what actually makes this all unfold. So we've got our first one here is DNA helicase, and that's what unzips the DNA molecule. It takes that double helix and splits it into two halves. And that split is called a replication fork. And again, I know you're going to have tons of questions about all these, but it's going to become much more clear when I start showing the diagrams. I just want to introduce the enzymes. Next, we have something called DNA primase, and that basically tells the next enzyme where to start making the copy. And then we've got the main player, the big guy, the most important one of the whole process, DNA polymerase. And DNA polymerase, that's the enzyme that actually reads, it kind of proofreads as it goes, and then writes the new DNA molecule. It's pretty amazing that it rarely ever makes mistakes. I mean, they happen, but it's super, super rare. They almost never make mistakes, and considering how many base pairs it has to create, it's quite amazing. And then lastly, we've got an enzyme that kind of glues the whole thing to get back together. It called DNA ligase. It's actually mainly it's really just on one of the new strands of DNA that needs to be pieced back together. So we'll talk about that in a little bit. So here we go. Here is our first look at what DNA replication looks like. So again, we've got DNA helicase here, and it comes in and unzips. So you can see here's the long DNA strand, here's the chromosome, and you've got this diagram shows this long DNA strand, and here as we get closer, it zooms in, you can see the base pairing, and you can see DNA helicase coming in, splitting it, and this is creating what's called a replication fork, like a fork in the road. You can see it takes two paths, and DNA helicase is what splits those base pairs. Now, what happens next is DNA primase, where did we go? DNA primase comes in and starts writing. Now, this is where it gets a little confusing. So DNA polymerase comes in. I said primase. DNA primase tells DNA polymerase where to start. So DNA polymerase comes in and starts writing the actual new DNA strand. And this is what gets a little bit tricky, because you can see DNA polymerase is writing nice and happily in this direction and can go on forever until it finishes. But on this strand, it's going in the opposite direction. So why is that happening? Well, again, we keep talking about this anti-parallel direction. So let's discuss that quickly, and then we're going to get back into the actual writing. So what we mean by that anti-parallel direction is that we've got these sugars, these 5-carbon sugars that make up part of that sugar phosphate backbone. And you can see that you've got an oxygen, and then all of these corners are carbons. There's actually a carbon right here too, where it meets the phosphate group. But look, on this side, the oxygens are on top. On this side, the oxygens are pointing down on the sugar, right? So basically what we're doing is we are counting the carbons and assigning them a number, and based on their position on the sugar phosphate backbone, we are going to name the direction that that side is going. So we've got 1 carbon here, 2 carbon here, the 3 carbon here, the 4 carbon here, and the 5 carbon here. And we add a little dash and say that that is the 5 prime carbon. So 1 prime, 2 prime, 3 prime, 4 prime, and then again right here where the sugar meets the phosphate group is our 5 prime. So you can see on this side we're going 5 to 3 and then phosphate group, 5 prime to 3 prime, which connects to the next phosphate group. Then the 5 prime, there should be a 5 right there. It's not there, but 5 prime, and then to 3, and in that fashion. Then the opposite, on the other side, we have the opposite. It goes 5 prime here to 3 prime to 5 to 3 to 5, and then ends at the end of this short segment, ends at a 3. So that's what we mean by anti-parallel directions, and that's where we get that 5 prime to 3 prime direction. I know that's a little tricky, but again, it's important to remember because DNA polymerase, remember our main enzyme, the one who does all the writing, can only write in the 5 prime to 3 prime direction. Okay, I'm going to say that again. DNA polymerase can only write in the 5 prime to 3 prime direction. This always confused me. It's not writing on the 5 prime to 3 prime strand of the original. It's creating a new strand in the 5 to 3 prime direction. Okay, you see that distinction? Here's a nice, really basic diagram of what I'm talking about here. Here's the original, the lighter blue is the original DNA that's been split by DNA helicase, came in here, split it, created this replication fork, and you can see that DNA polymerase is writing 5 prime to 3 prime along the original 3 prime to 5 prime direction strand. And then on the opposite side, we're doing the opposite. So since DNA polymerase can only write in the what? Yep, 5 prime to 3 prime direction, it has to write along the strand that's going the opposite way, right? So you can see this strand is going 5 to 3 in this direction, so it needs to write 5 to 3 in the opposite direction. Do you see this? So what we do is we name these 2 strands. Oh no, we went the wrong way. We name these 2 strands. We've got the leading strand, which is the original strand that DNA polymerase is able to just write indefinitely on. Okay, it can just keep going, no problem, can just keep going towards the direction that helicase is splitting. And then we've got something called the lagging strand. What the lagging strand is the strand that is going in the opposite direction. All right, so the lagging strand has to write in segments. It can't just keep going because eventually it runs out of real estate down here. So what it does is DNA primase lays down a little RNA primer. These red dots, these pinkish red dots represent an RNA primer. It's just one little segment of RNA, and then polymerase writes, and then it stops where DNA primase has set another RNA primer, and then you get another segment here, and then it stops where it meets that next RNA primer that primase laid down, and then DNA polymerase writes along the next segment, and it goes in that direction until it runs out of room.